processing by neurons, and we're starting off with this picture here, which is a picture of many different kinds of neurons. And so you can see that they come in a variety of shapes and sizes and, of course, functions. And a huge question in neuroscience has always been, how does the structure of a neuron relate to its function, its signal processing that it does? So you'd be right in thinking that a little neuron like this with a wispy dendritic tree might be doing something very different than a neuron like this with an amazingly significantly branched dendritic tree. These pictures aren't to scale, incidentally. That is to say, the size of this neuron is not roughly half of the size of this neuron. These pyramidal cells are large cells. They're large neurons, and so they have very extensive dendritic trees. And naturally, if you consider where inputs go on these neurons, there's huge numbers of inputs landing on very specific structures within these dendritic trees, and these granule cells have much fewer neurons. And so the big question is, if we consider where synaptic inputs go on a neuron and what the shape of the neuron is like and the nature of its dendritic tree, what kind of calculations can a neuron be doing, and how do these things relate to the function of the neuron? So this, of course, has been studied for a very long time. There we go. And I always like to begin this saga with a story about the sociology of science. And it involves two scientists, and the first one is Sir John Eccles, a very famous British neuroscientist. He won a Nobel Prize for his chemical theory of synaptic transmission. And so he was a very well-known neuroscientist. And in this book, The Physiology of Synapses, he made this statement: synapses on dendrites are virtually ineffective if situated on the more remote regions of the dendrites. Well, okay, so that's saying something quite important. I just said that on this big pyramidal cell, lots and lots of inputs go out in these tufts. Sir John Eccles is saying synapses on dendrites are virtually ineffective if situated in the more remote regions of dendrites. So he believed that the synaptic inputs that really affected the responses of neurons were those landing near the soma or on the soma. Well, where did this come from? Where did this idea come from? It came from what I like to call his mental model of how neurons function, right? He wasn't backing this statement up with a deep, thoughtful, mathematical, let alone computational analysis of how neurons function. It was his intuition. And I believe that biological systems in health and disease are so complex that our mental models fail us most of the time. That's why you're engineers studying biomedicine, because you want to bring your engineering perspective into biomedicine. So you want to think about a question like this in a really quantitative way, in the way an engineer would think about it. Well, his student, Will Rall, thought about this problem like an engineer would think about it. And Will Rall, as I said, I believe was a postdoc with Sir John Eccles in Australia, and then he moved to the National Institutes of Health, where he set up the mathematical research branch of the NIH. And he ran that for many, many years. It's still active. Today it's run by Arthur Sherman. Will Rall was a wonderful person, great guy. However, he differed with Sir John Eccles. He asked the question, who says synapses need to land near the soma to have an influence on the firing pattern of a neuron? I want to answer that question quantitatively. So he started using mathematical modeling to approach this question, as an engineer would. And he developed what we now know as the linear theory of the neuron. It was a linear systems model of how dendritic signal processing occurs. And it was used to address the question of whether or not distant synaptic inputs could in fact influence the firing pattern of a neuron. And so this is an interesting quote from Will Rall. And so he presented this, what he called the biophysical mathematical theory in computations about the nature of dendritic signal processing in neurons, and he submitted it to the Journal of General Physiology. And there was this obnoxious reviewer. This was back in the, you didn't get your reviews by email in this day, right? There's no email, right? So you got the paper back, and there were margin notes in the paper handwritten by the reviewer. And he recognized somebody's writing, right? And the nature of somebody's comments. And it was Sir John Eccles, right? So Sir John Eccles ripped him to shreds. He said, this is just foolishness. This is silly stuff. And he recognized that. He knew this. This bothered him tremendously throughout his entire career. I confess that I felt rather depressed by the rejection. Well, welcome to the club, right? We've all felt depressed over rejections of papers. But this was a struggle between Sir John Eccles and Will Rall for many, many years. And the thing that bothered Rall was that despite the fact that Rall overwhelmed Eccles with a mathematical theory and proof that distant synaptic inputs could matter under certain conditions, because he built a mathematical theory of this, Rall would never accept it. Never said, you're right. And what made matters worse is that Eccles was Rall's postdoc mentor. And so this really got to him over the years, and he never let go of this resentment. I mean, the fact that he would say this in his autobiography is kind of telling. So it's an interesting story of the clash of two brilliant people. They were both brilliant people. But Will Rall gave us the modern theory of a neuron. And so that's what we're going to talk about today. And here's my simple cartoon model of how Will Rall thought about this problem and went about solving it. So here's a stick figure model of a neuron. I haven't drawn the soma, but let's think there's a membrane enlargement right here that's the cell body of this neuron, and there's an axon running off here, which is where action potentials are generated. And synaptic inputs land out on this dendritic tree. And I've just drawn a stick figure representation of this dendritic tree, right? And so, you know, sometimes models come along, they're wrong. Fundamentally, Will Rall's thinking about this was wrong. But the models got to the essence of the problem and allowed him to reach a very fundamental conclusion, even though many of the assumptions of his modeling were in fact wrong. We know today it's wrong, but it doesn't mean that nothing great came out of this modeling. It did, and here's what came out of the modeling. Will Rall said, I'm going to start off by forgetting about all of this branched complexity that we saw in the last picture of real neurons, all of those different neurons. They're very complex structures. We're going to think about the world's simplest neuron. It's a cell body attached to this big long tube, and that's the dendrite. And so this is just a membrane cylinder, and it has some length, and it's got some diameter. And I'm going to think about synaptic current, the consequence of there being a synaptic input far out on this dendrite. The way I'm going to think about it is that synaptic input just injects some current, and we're going to call that I synaptic of T. It just injects some current into this tube, and so what happens to the charge that's injected? It begins flowing along the fluid-filled center of the tube, right? That charge propagates down the tube. If there are ion channels in the cell membrane, some of that charge leaks out from the cell membrane. So some of the charge that's injected here gets lost as it goes out through the membrane. And by the time that bolus of charge reaches the soma, depending on how much was lost, this synaptic input produces a large or a small voltage depolarization at the soma and may or may not cause the neuron to fire. Yes?
Just to be clear, this is channel independent, right? There are no like ion channels in this model?
Very good question. In Rawls' formulation, there were ion channels for ions to move across a membrane, to leak out of this cable. They have to go through something. They aren't going to go through the hydrophilic membrane, hydrophobic membrane. But they're going to leak out through channels. But Rawls' core assumption is that we're going to think about the current voltage relationship of those channels as being completely linear, purely resistive. So there are channels, but there aren't voltage-gated nonlinear channels. And that's what I meant when I said we now know there are some fundamental flaws in the way Rawls thought about these things. We know that there are voltage-gated channels in neuronal dendrites. We know that. It's been proven time and time again. And so that makes, in some sense, the theory wrong. But even though those voltage-gated channels are there, there's still an essence that comes out of the theory that's right. And that essence has to do with how does the morphology of this dendrite influence the amount of charge that gets to the soma. Fundamentally, even with voltage-gated channels, we can do fancy things like argue about small perturbations of voltage around an operating point and linearize these models and get all fancy. And that allows us to leverage the kind of thinking that Rawls had. So this is the fundamental question he asked. You inject charge out here from a synapse, how much gets to the soma to cause the neuron to fire? That's the key question. And he answered it with mathematical models. Now just to say, okay, so one of the problems is people like me come into systems bioengineering true without knowing the full context of everything you've studied up until this point. Okay, I confess, it's true. So do you know what a synaptic input is? Do you know what a synapse does? Do you know chemical synaptic transmission? You've studied that, right? How about EPSP and IPSP? Have you heard those terms? Excitatory postsynaptic potential, inhibitory postsynaptic potential, right? Okay, yeah. So that was a yes. They've heard them. Okay, so let's talk about that in reference to the circuit here. So this circuit here represents a little subcylinder of this big membrane cylinder, just a little chunk of it. And so the membrane, the surface of that little compartment, has some capacitance associated with it. The bilayer lipid membrane has some ability to separate charge, and that's captured by this capacitance element. And it has some conductance associated with it, and that conductance is in series with a battery that represents the reversal potential of the ions that move through that conductance. Are these terms ringing a bell? Reversal potential. So that's what this is, a reversal potential. And so a synaptic input, the effect of a synaptic input is to increase this conductance. But whether that produces an excitatory or inhibitory postsynaptic potential depends on the reversal potential of the ions that move through that conductance, right? So if that reversal potential is less than the resting potential of the cell, then the cell membrane potential will be locally hyperpolarized, producing what is known as an inhibitory postsynaptic potential, a response that counterbalances other excitatory influences to this neuron. Whereas if this reversal potential is greater than the resting potential of the cell, changing its conductance will cause a current to flow into the cell that excites, that depolarizes the membrane potential. So that's the fundamental difference. Very good. Your questions helped me work through all of that. Thank you. Okay. So the simplification that Rawls did fundamentally in his first paper was, let's forget about this branching pattern. Let's just think about a dendrite that's a tube. to make life even simpler, we're going to keep all the properties of this tube the same everywhere. The specific membrane resistance, capacitance, length, it doesn't change. I'm sorry about my text messages. There we go. That's done with. And so he simplified the problem as much as he could. Now, in so doing, he was able to work out closed-form mathematical solutions for, let's go back, not just the problem of, given a current injected here, what's the distribution of voltage along this membrane, and in particular at the soma? Not only did he solve that analytically, mathematically, pencil and paper, but analytically, he could also solve the problem of injecting current out here on the dendrite of this complex branch structure, and the equations that he derived would allow you to calculate, pencil and paper, the distribution of voltage everywhere else in that dendrite, including at the soma. So it's kind of a remarkable mathematical theory. Yeah, I know there's a lot of assumptions behind it and there's simplifications, but the idea that you could look at a neuron and approximate its structure as a stick figure like this, and on pencil and paper calculate the influence that a certain amount of charge injected into the neuron anywhere in the neuron would have everywhere else, that's a remarkable contribution, and from that emerged what we call the modern theory of the neuron. So before we get into that theory, I just want to say a few words of how do we approach these problems today. Well, okay, here's a model of a neuron and a dendrite. And look at this branch here. It's broken up into 3 subcompartments, 3 short tubes connected together. So here's a branch, and notice that the branches are getting smaller in diameter with branch order, right? So that typically happens. And so these branches are themselves broken into small tubes, and they're connected in series. And we make the tubes small enough so that, to a first order of approximation, everything is equal within that compartment. So concentrations and voltages and such are approximately the same in that compartment because it's so short, there's no difference in voltage here versus here. And the way we get these representations, this would have been around 1980, 1990. The way this would have been done is you look at a neuron under a microscope, and you've got a projection tube coming off the top of the microscope, and it projects an image of the neuron onto a piece of paper. And what did people do? They literally traced with a pencil the outline of the neuronal dendritic tree on that piece of paper. And by knowing the field of view, the magnification, they could get a distance metric, and from that piece of paper tracing, measure dendritic lengths and diameters, and do this manually, decompose that neuron into a bunch of small compartments, one by one, individually, getting the length and diameter of each compartment, and that sets its electrical properties. We'll see that in a moment. And then you could on computer simulate the response of a fine scale representation of this dendritic tree to any kind of input you cared to apply. And then, more recently, along comes super high resolution imaging methods, multiphoton imaging methods like you see here. So here's, I think this is a cortical pyramidal cell. And you see that it's reconstructed using a 2-photon imaging system, which has a resolution down to like 500, 600, 700 nanometers. And so the reconstruction was done in an automated fashion by optically sectioning the specimen and then using digital image processing methods to delineate where the dendrites are and to figure out their sizes all automatically. Look here, we're zooming in on one of the dendrites. Okay, there it is, and you see these small structures coming off it. They're called spines. Anybody talked about spines yet? No? All right, let's talk about them. Here's a spine. Look at the shaft of that spine. It's tiny. It's extremely, extremely narrow. And it enlarges at the heads. In these kinds of neurons, this is almost always where the synapses go, is right on the head of that so-called dendritic spine. These ultra-fine, small-scale structures, they're the sites of synaptic inputs. And we now know that the dimensions of these spines can change. And they can change fast. They literally twitch. You can image them moving. They've got actin filaments in them. They move. And so it turns out that this is a way of adjusting the synaptic weight associated with that input. And I think we'll talk about that in one of my coming lectures. But the message of this slide is nowadays we have super high resolution methods for building these models of neurons and automatically finding out where the dendrites are. You can see these nice fine scale reconstructions of not only the dendrites, but these ultra small spines. And we can automate the process of constructing computational models and simulating the electrical behavior of even big dendrites at very, very high resolutions. That's how it's done nowadays. But at Will Rawls' time, the theory of what to do with this data wasn't even there. And so he's the one that created this theory. So, I like in lectures to tell you what you need to know. I've got 2 slides here. That's the bottom line of what you need to know from this lecture. I'd appreciate it if you didn't go to sleep for the rest of the slides, but yeah, this is the bottom line. So here's our dendritic cable. It has some length. And this capital X here, that's going to represent, as we will see soon, a normalized length. It's not length measured in microns or millimeters. It's a dimensionless length variable. Anyway, this dendrite has some length. It's got some fixed diameter. It's got this terminating conductance at the end, at position capital L, which I call G out. Now this is often confusing. What the heck does Winslow mean by G out? All right, well, that's the output. If we are sitting here at this point in this electrical circuit, at this structure, and we're looking out the end of that dendrite, that's the conductance of whatever we're looking into. So maybe this neuron branches here. Maybe there are additional dendrites out here. We're sitting at this point on the end of this dendrite. We're looking into the remaining branch structure. G out is that terminating conductance looking into that structure. Or maybe this is the very end of the dendritic tree. It just stops. And this diameter is so small that the surface area of that face is so tiny that it's sensible to assume there aren't any channels there. There's no current out of that end of that dendrite. G out is 0. That's called the sealed end boundary condition. It figures in Rawls' theory very heavily. So note that, sealed end boundary condition. G out 0. It's a simplification. But that's the intuition into which, behind this terminating output conductance. If you don't get what that means, raise your hand and let me have another shot at it. Does that make intuitive sense to you? It's just the ratio of the current leaving this node divided by the voltage at the node. That's by definition the terminating output conductance. So that current is 0 if this face is small and this neuron isn't connected to anything else. If that neuron is connected to other portions of the dendritic tree, there will be some non-zero conductance looking into that terminating structure. So what Will Rawls showed was if we have some intrinsic membrane properties of this cable and we know its length, we know its diameter, and we know this special terminating conductance, we can figure out voltage everywhere along the cable in response to anything. So he did that by deriving the cable equation. You've got to know the cable equation. Right? So V here is voltage. What is voltage? I mean transmembrane voltage. Let's think of the environment within this tube that's intracellular potential, it varies in space. It can vary in space. The voltage outside that tube is that of the extracellular space, and so this V is the voltage difference across this membrane. In the Raleian theory we're going to develop, we're assuming that this extracellular space is at a common potential everywhere. It's ground. That sets ground in our electrical circuit. It doesn't need to be that way, but it's a simplification. So voltage is the potential difference between potential inside the cell and outside the cell, and a voltage exists for every location X. A voltage exists as a function of time, and capital T here is again a dimensionless time variable. We're going to take real time and normalize it by an important quantity that is in units of time, and so we're going to get a dimensionless variable. So both of space and time are expressed in terms of dimensionless variables. That's very important. We're going to talk about the input conductance of a semi-infinite version of this cable. Okay, so I set this up saying this cable has a finite length. Well, suppose L goes to infinity. This cable runs off to infinity. It's infinitely long, and I'm looking in at the start of the cable. So the input conductance of this cable, if I apply a current at this point, and it produces a particular voltage, their ratio gives me the input conductance at that point, right? And so this thing called G infinity is the input conductance of an infinitely extended version of that cable looking into that end. And so G infinity and G out are very important cable properties. And it turns out that if I know the radius of the cable and I know another quantity which is the axial resistance, the resistance to current flow per unit length along the cable, capital R sub I, there's a relationship for this. We know the input conductance of a semi-infinite version of this cable. It's pi r squared over r sub I times this thing called lambda. It's another cable property. So we'll figure out momentarily what lambda is. We'll explain what r sub I is. R is just cable radius. And so given these fundamental cable properties, we can get these two quantities, and armed with these two conductances, we can figure out whatever we want to know about this cable. That's what Rahl showed. So you got to know that. You got to know that, and we'll derive it. Now, what about the input conductance of not a semi-infinite version of this cable, but the finite length version? We'll haul it back from infinity and make it have distance or length capital L. Well, this is the input conductance of such a cable. And it's expressed in terms of the G out, the G infinity, and the dimensionless length of the cable, and we'll derive this relationship. I wouldn't be telling you these things if you didn't need to know them. They're important to the Rahl theory. And the reason that they're important to the Rahl theory is that if we want to know, at least in the steady state, if we apply a current anywhere along this cable, let's say right here, armed with these things, we can compute the steady state distribution of membrane potential anywhere along that cable, and it's given by this formula here. So that's why you have to know G in, understand what G out is, know what G infinity is, because they're all involved in calculating the voltage in response to a maintained current anywhere along this cable. We'll let time go to infinity, right? We're just looking at the steady state. Things get a lot more complex when the system isn't in the steady state, but there is a Rahl theory for that. And if any of you are interested in the Rahl theory for that, I've got this document called The Tome, which is like 100 pages long, and I wrote it up on Rahl cable theory. And it's on Blackboard, I guess, and you can dig into the tome to your heart's delight, and you can read this theory all you want, and then we derive how to get transient responses along this cable in response to just about any kind of input. But all I'm asking you to know are steady state responses, because that's enough for our intuition to really drive our intuition about how neurons work. So that's what you need to know. Yeah?
Are we assuming that when the current is entered into the system, it can only move in one direction? So like you're only putting the current into one end?
Yes. Or at a point, or at this end, anywhere. What we're really assuming, what your question is driving at, is how do we put current into a neuron? We stick an electrode in it, right? And so the assumption here is that when we poke a hole in a neuron with a glass microelectrode, there's no leak of charge out of that neuron through the hole that we poke in it, right? And so that's not strictly true. There could be a resistor here that runs to ground. And so not all of the current that you inject down the electrode will necessarily make it into the neuron. Some of it might leak out into the outside world. And that is just a matter of serendipity, how well this glass microelectrode impales the cell. If we're doing this thing called patch clamping, does that ring a bell, patch clamping? Okay. You know what it is then. You know, you take a big bore glass pipette and you put it up on a cell and apply negative pressure, and the membrane ruptures and adheres to the inside of the pipette. That makes what's called a giga-ohm seal. The special property of patch clamping is that it essentially reduces this leakage resistance to 0. So we're assuming 0 leakage resistance here. That's an assumption. Yeah?
How would you account for multiple inputs in this model?
It's going to be linear, so we use superposition.
All right.
This is a linear model by construction, so it's the superposition method. Was there a hand up over here? I thought I saw something. Okay. Very good then. All right. I mentioned this case. Now, right, here's the equation for voltage. Well, what if the sealed end boundary condition holds? What if the output conductance is 0? Well, this term goes away and this term goes away, and therefore we get a tremendous simplification of our expression for voltage. That's why it's a special condition in the Rawl theory of the neuron, because it leads to dramatic simplification here. So what I want you to know from this lecture are these equations and how to apply them. There's an important way in which we're going to apply them. And their derivation. Know about their derivation. And here's the important way how we will leverage these equations. These insights from Rawl. Remember, I said that he had a theory of how to calculate, pencil and paper, the distribution of voltage everywhere, even in a complex neuronal dendritic tree, in response to a current injected right here, let's say. Maybe the question we're asking is, what effect does the charge that we inject out here have on the soma? How effectively can it drive this neuron? You can answer this question using Rawl's theory. And the way you do it is the following. Remember, from that previous slide, I said that if we inject current into this end of a dendritic tube, all we need to know are some intrinsic membrane properties, its length, and its terminating conductance. Right? That's what I said. Then we can get voltage everywhere along that tube. What's the terminating conductance right here? If we're sitting at the end of that tube looking out, it's this input conductance plus this input conductance. Okay? Does that get us anywhere? Yeah, it does. We've got an equation for, I just showed it, for this input conductance, so we know that one. But what about the conductance looking into this big structure here? I didn't give you an equation for that one, but there is an equation, and it can be figured out iteratively. So how do we do that? So let's think it through for just part of it. Let's forget all this stuff down here. Let's say this was the structure. How do I get the input conductance looking into this port of the dendrite? Well, I march all the way over here Maybe there's a sealed end boundary condition. Doesn't matter if there is or isn't, as long as we know what the terminating conductance is. We know the input, we have an equation for the input conductance of that tube. We have an equation for the input conductance of that tube. The input conductance of these two together in parallel is their sum. If we have the input conductance looking into these two structures, we now have the terminating conductance on this tube. Right? So if we have the terminating conductance on this tube, we can calculate its input conductance. And in fact, since we've removed the rest of this dendrite, this is all one big tube now. So we've got its terminating conductance, and we know the input conductance looking into this node. So we know this and this. We can figure out voltage everywhere along here. Let me pause. Does that make intuitive sense? Do you follow me? I mean, it's just iteratively applying the formulas that I showed on the last page to show you the method that you can use to compute voltage anywhere in this dendritic tree. So it allows us to compute voltage in response to this input everywhere along that dendrite. And now we just keep repeating this. We've got the voltage here at this termination point. Maybe that's the sealed end. We've got that terminating conductance, whatever it is. We've got voltage everywhere along that dendrite now. And by using the method that I just described, we now know the terminating conductance here, so we can get this voltage, and so on and so forth. You just iteratively sweep through the dendritic tree doing these calculations over and over. You can, on pencil and paper, get any voltage. This is the linear theory of the neuron. This is the Rawl single neuron model. And what did he show with this theory? He showed that depending on the membrane properties and the length and diameter of these dendrites, distant inputs can have significant effects at the soma. Distant inputs can integrate together to have a large input on the soma. If you've got a bunch of them going far out in the dendritic tree at the same time, they can sum up and influence the soma significantly, causing this neuron to fire. That was different from the Eccles view of the neuron. This is the big mathematical modeling contribution of Will Rawl. Okay, so let me keep track of time here. When do I have to stop? 2:30, 2:20. Okay, well. Yes.
Why is there representation of the injection of current on your right branch rather than an injection of voltage? So why is that synapse having an injection of current rather than
Right. Physiologically, the earliest technique to be developed for recording in neurons was to use very fine-tipped glass microelectrodes that can poke into the neuron, and they can sense voltage or they can inject current. Neurons are large structures, and so what you can't do with that technique is perform a voltage clamp, because the neuron is so spatially extensive that it's impossible to lock different regions of the neuron to all of the same potential. So it's really impossible to do a voltage clamp. And so what you can do is inject current at a particular location and measure voltage elsewhere. That's what's measurable. It's very difficult to lock voltage to a specific value in a single neuron because of this inability to do space clamp. But it's much easier to get a neuron, a microelectrode onto or into a neuron and inject current and look at how it influences that neuron. So that's one reason. The second reason is that the effect of changing a synaptic conductance is really to produce a current flux into the neuron. That's the fundamental thing it does. When that conductance goes from 0 to something, there's now an electrical driving force across that channel. It has some permeability and charge will move through the channel. That's like injecting current. And so what a synapse really fundamentally does is inject charge into a current. It's as if you were injecting a current directly into the cell, only the way you're doing it is causing a membrane conductance, some set of ion channels, to open. to conduct that current. Yes?
Could you go to the last slide,
Mm-hmm.
So for that red segment, would G out be where the BI is or where the P is?
It's both of them. We're kind of sitting at the end of that dendrite, looking into the world that terminates that dendrite. And what we see is this tube, and we see this tube. And so the output terminating conductance here is the input conductance of this tube plus the input conductance of this tube and everything else that's attached to it. So this input conductance to this tube was easy to get because it's just a single tube. This is a little harder because we have to iterate our formulas over and over to figure out what the input conductance is looking into this tube with all of these other structures. But fundamentally, the terminating conductance here, the G out, is this in parallel with this. So they just add. Yeah. Alrighty. Okay, here are the definitions we're going to work with. We've already talked about capital R sub I, specific axial resistance, resistance to current flow in the axial direction of a uniform cable, 1 centimeter long, end face surface area 1 square centimeter, a special chunk of cable. And it will have units of ohm centimeters. And in a moment, we'll see how these units make sense. We're going to work through the units and make sure that they are sensible. That's the units of axial resistance. Specific so that's the resistance to current flow looking down the core of a tube along the axis, axial current flow. Specific membrane resistance, take a dendritic cable, unfold it. It's got some surface area. It's got some density of channels, some number of ion channels in that surface area. That net resistance to current flow through all those channels is known as specific membrane resistance. That is the resistance of a patch of membrane with surface area 1 square centimeter. So the units of this are ohm centimeter squared, right? And then finally, CM, capacitance. You take the surface of a tube and unfold it. It's got some surface area. That tube membrane is really a bilayer lipid membrane. It's got some ability to separate charge. So it's by definition a capacitor. And this is essentially a biophysical constant. Pretty much all biophysical bilayer lipid membranes have a capacitance of 1 microfarad per centimeter squared. Right. Okay, so these are 3 physical parameters of a cable. This is really determined by the diameter of the cable and how much fluid there is to support charge conduction. It could be affected if there are organelles or myofibrils within the dendrite. If they take up space and they obstruct the movement of charge, they will affect this. But in Rawls' world, we're going to estimate this, and it's going to be a number, and it's applied in his model. And RM will depend on the density of channels in the membrane. This is a biological constant. All right, so we're going to start off, we're going to derive the cable equation. Not today, but ultimately. So don't leave yet. I didn't say anything. So we're going to do this by taking a cable and breaking it into many little subcylinders. And to get the electrical description of each of those subcylinders, we've got to use those fundamental membrane parameters that I just talked about. So here's a little cylinder of length delta x, diameter 2r. What's the axial conductance of that? It's pi r squared over delta x, the length, times 1 over ri. All right? So this is the surface area. So it makes intuitive sense that the bigger that surface area is, the more charge is readily conducted down the core of this conductor. So it makes sense that the conductance increases with that term. The longer it is, the greater the resistance to moving charge out of the other end of the cable is, so it makes sense that it's inversely related to length, and that multiplies the intrinsic cable axial conductance. So let's do the unit thing. This is centimeter squared. This is centimeter times 1 over ohm centimeter, right? So the centimeter squareds go away, and we're left with 1 over ohms, which is Siemens. That's the appropriate unit of conductance. And I guess I have to stop here. We'll pick up and go through the other cable parameters and derive the cable equation and then solve it in the steady state next time.
I have a question about the dendritic tree. So this is the dendritic tree, and then when the signal passes through the pathway toward the soma, does it flow only in this direction? How come it doesn't flow to the other direction?
It does.
So it can potentially inhibit other signals.